You ready?
So, good evening, everybody. Thank you so much to Mandy, Josh, and the White Sutton Foundation for inviting me and to allow me to represent Simon Searchlight. I want to start by giving you a little bit of background of what Simon Searchlight is in case you're not familiar with the study. We are an international research study that has a goal of generating resources for research. So more than doing first-hand research ourselves, we get 100% of the funding to create a natural history study, collect clinical data, collect biospecimens, create resources. Thank you. Provided I don't destroy the place, okay? Sorry. Well, let's try this again. So we provide resources based on medical and behavioral histories. We collect biospecimens. These are optional blood samples, so it is not a requisite to be part of the study to donate blood. However, they're useful samples for sure. And once we have enough information about a specific gene, we start collecting that information and returning it to families. And we do this in many different ways. It can be through quarterly reports where we show what new information we have. We attend meetings like this one. We have Facebook groups. We have different printed materials. Gene pages that you can access online with information. We have genetic counselors that can help answer questions. So part of the goal is to get back to the family. A new feature that we're hoping very much to be able to release this upcoming year is a data visualization tool where instead of just getting static data that we provide you, you can do searches of our data. So you can go and ask your own questions, or if you're partnering with a doctor or a scientist, you have, say, a pediatrician that is not super familiar with this, they can go in and do that, and everything will be, again, broadly accessible, completely de-identified, so you nobody else knows who you are or your child is, and free. And other things we do is we share samples and data with scientists. We can also connect scientists with the families, and I'll go into a little more detail later. And we have scientific publications with the goal of advancing the knowledge in this disease. So, okay. Yes. So I don't expect you to be able to read any of this, but just so you know, we have more than 185 genetic conditions in the list of the conditions we investigate. And this can also be important from a different point of view. Sometimes there's strength in numbers, and researchers are thinking, okay, which rare genetic conditions share, for example, I don't know, and unusual neurologic complications such as tethered cord. They can go and look through across all these conditions and try to see if there are common mechanisms in different diseases. Could that lead to a shared drug, for example? Why would we want to do that? Because a shared drug would be more profitable for pharma and therefore maybe more feasible to get moving. So there are reasons for which we want you to know that this is not a single condition, it's many. And we currently have materials in all those languages that you can see in the box here with a blue square. Not all the instruments are yet online, ready to be used. However, they have all been QC'd and translated. So 2026 is a year where anyone can participate with 100% of the surveys in those languages. We collect information about demographics, genetics, medical history, seizures, sleep problems, development, and behavior, both in-house instruments and also those that are created by other publishers, and some of them that the FDA requires to even consider any clinical trial. Sorry. Some of them are collected. I'm having a little bit of trouble. Okay. Some of them are collecting only once, some are on an annual basis with a goal of seeing what is the natural history, how does this condition progress over time. And then we generate these reports where we summarize that information for the families. I mentioned that there's also a way of doing a little bit of matchmaking between scientists and families with specific conditions. We call that research match. You could almost think of it as dating, but for these conditions, because what happens is a researcher approaches us and says, okay, I have this study that I really, really want to do, but I don't know where to find people to recruit them to participate. So what we do is we look in our database if there are people with a specific conditions or requirements that scientists is looking for, and then we send out emails or letters to all those people that meet the criteria, and then you can pick and choose what you want to participate in. Furthermore, we often fund the researcher. If they don't have enough money to do this, the Simons Foundation helps them with the funding. And also, if they, for example, don't have enough funds or technical ability to create the surveys online, to obtain new samples or anything like that, we also have a platform to do that. So you choose what you want to participate in, but it's a way of connecting you with scientists. The results are that so far more than 120 scientific publications have come out of Simons Searchlight, and these are very varied in scope. It is from basic science to pure biology to behavior to imaging of the brain to thesis for new scientists who are just training and getting involved in this realm. And for scientists, if they want to access just data and materials and not directly contact or involve the participants, they can reach out to the Simons Foundation through what is called Safari Base. And in Safari Base, they submit an application for what materials they want. Again, everything is shared with the scientist based on what you consented to and without any identifying information. And it usually takes up to a maximum of 2 weeks to get a response from Safari to the scientist about providing them the materials. Again, materials provided to them at no cost. So that is the commitment of the foundation. And when we want to talk about specifically white Sutton and abnormal variants in the POGZ gene, this is where we are at with the people that have participated. So far, there are 55 families that have signed up online. So the first number where it says step 1, that refers to families. So it's at a minimum 1 affected person per family. There are very rare occasions when there's more than 1, but that happens. And the next step after signing up is to provide a genetic lab report. We need that to confirm that you have the condition and that we have sufficient information to include you in the study. If you, for whatever reason, don't have a good genetic report, we do offer retesting if needed. So maybe were tested long ago and the quality of the report is not very good, or at that point it was a variant of unknown significance, our genetic counselors review that too. If necessary, there's retesting, there's cascade testing of the family. So this is important to know. After that has been reviewed, we ask for your medical history. 35 people have gotten that far. 35, 36 basically have started filling out surveys. 17 people have donated samples, so 17 carriers of mutations. There are additional family members that have donated samples as healthy controls, but 17 carriers. And many of the people in your community are already in the longitudinal part of the study, so every year contributing more information. Now, these numbers include genetic variants of pathogenic, likely pathogenic, and also the unknown significant. And why is that? Because the technology and the knowledge is advancing so quickly that every year we're able to reclassify many of those variants of unknown significance. So if there's anyone here, and I see heads nodding back there, we'll talk after this and we can try to brainstorm about how to reassess. And maybe this is not the year, but you have to keep trying, okay? So next, this is a schematic of the gene. This is a POGZ gene here at the bottom. And every little, as Dr. Wendy Chung would say, little lollipop up here is the site of a specific variant. In the case of the gene, you can see they're spread all over the gene across the whole length, and there's great variety of abnormal variants. However, something that is pretty common are these that say FS and then have a little asterisk, and those result in a truncated protein. Those are usually more severe variants than those that just change one letter in the DNA. Those mutations with little asterisks means that once the machinery in the cell starts reading that gene, it stops at that point and it doesn't progress. So obviously those that stop at the very beginning of the gene are expected to be more negative in terms of impacts than the ones that just miss the very tail of the gene. So that's why you see so much variability in the clinical features. We know of families that are in 83 countries. Well, no, this number is wrong. I checked yesterday, sorry. Simon Searchlight has participants in 108 countries as of yesterday. We just have 2 in Africa, so that's super exciting. And now 37 of those families are families with variants in POGZ in 7 different countries. The darker the color, the more people that are in that specific country. So as expected, since this is where we are based, the US is the country with the most participants. So there are 28 participants living in the US. Now, in terms of what specimens we have in our biorepository, I said there were 17 people that had donated specimens. Some of them we have run out of. So they have been requested by some scientists and we have run out of specimens. So currently we have specimens derived from 12 people. 7 are whole blood DNA, 5 are lymphoblastoid cell lines. And for those of you who are interested in knowing about iPSCs, which are the pluripotent stem cells that are now being used to create models of the disease in a petri dish, The Simons Foundation also sponsors the creation of iPSCs. This is a lengthy and costly process, and they're working on 5 specific variants. The variants chosen for creating the cells are based on scientific need. There's not enough funding to do every single sample, every single variant. So based on those that scientists have pointed out as crucial right now, those are the 5 lines that are in development. They're not ready for distribution yet. It costs many thousands of dollars and about a year of work to generate one single cell line, but scientists can obtain them for a nominal fee that just covers the shipping, so $124. They just have to apply and justify how they're going to use them, so it's worth it, so they're not wasted. Coming up next year too, this is also kind of hot off the press, PBMCs, which are the derivatives of blood that are used to generate these cell lines, will now be made available. So if scientists have either the money or the technical ability to create the iPSCs themselves, they can just request them, and then we're not so limited in the number that somebody can create. In terms of medical history data, we're showing pathogenic and likely pathogenic here. So we have complete medical history information from 27 individuals. If you look at this graph of the ages, you have on the horizontal axis the ages in years, and in the vertical axis the number of people, and you can see that unfortunately we have very few adults. So it's basically one adult in each of these subsets, and that is one of the reasons for which we don't know very well what happens in the long term. Now, why is that the case? Because these adults were diagnosed with a neurodevelopmental disorder when they were children, but there was no genetic testing. And even if there was genetic testing, the gene wasn't even known. So one of the goals for next year that we're going to try to work on is to try to start finding those adults. And the Simons Foundation will likely cover the genetic testing for anyone who's an adult that has an undiagnosed condition, to see if we can deepen our knowledge. It's not that they're not there, it's that they don't know and we don't know. So what are the main conditions we observe? Again, these are development and behavioral conditions. Always your denominator is 27 people were asked. So out of those, the most common is language delay or impairment in 85%, intellectual disability in 93%, 48% with autism, and then there are a few other more behavioral conditions that are relatively common. And I'll be happy to share the slides later if anybody wants. Here are other non-behavioral issues. At the bottom, you can see that neurological, gastrointestinal, and vision are the most common. Neurological is 74%, gastro 70%, vision 70%. And then others, I'll go into a little more detail of each of those. So here is the highlight of the neurological issues. Out of the 27, 20 individuals have neurological problems, that's 74%. The most common being low muscle tone in 67% of people, and a smaller head size in 48%. Other things that are common are coordination problems and changes in muscle tone. 5 out of those 27 have reported seizures, so that's 19% of all cases. There's very little granular data because there are only 5 people, and making generalizations out of 5 people is problematic. But what I can say is that 20% compared to other neurodevelopmental disorders is relatively low, so that's good news. Gastrointestinal, and that is also very common. 19 out of 27 had different problems. gastroesophageal gastroesophageal reflux disease and constipation being the most common. This can be severe enough that some children really have problem eating, gaining enough nutrition, and it can lead to the need for G-tubes or other supplementations of their diet. The Nissen fundoplication is the surgery to correct the gastroesophageal reflux, so those two go hand in hand. As to vision, that is the other common problem. The most common problem is strabismus, which is crossed eyes, but there are other different alterations, and this up here where it says surgery to the eyes most commonly is to correct the crossed eyes. Other slightly less common are endocrine. It should say endocrine, not neurological, but 11 people had endocrine problems, the most common being problems managing their weight. But they can also have the opposite, failure to thrive, some diabetes, short stature, and in some cases, early puberty. Less common, fortunately, are heart problems. 5 people had heart problems, and they vary by type. So it can be high blood pressure, rapid heartbeat, or some congenital malformations that required surgery. A few orthopedic problems, scoliosis being the deviation of the spine. Pronated feet are feet that are turned inwards, and pectus carinatum is when the chest is kind of pointy, so the sternum tends to point outwards. And finally, there are children that have had other surgeries that were not described earlier, so head and neck, mainly tonsils and adenoids, and PE tubes for their ears, and a couple of other rare conditions or surgeries. In terms of medication, there are several people that require medication. These are mostly to help with their gastrointestinal problems, so to help with the motility of their GI, be it to reduce the GERD or to prevent the constipation, and some other ones to help with sleep, mood, and behavior problems. However, we asked if there was any specific medication that people found particularly effective, and there wasn't a single one that really made a big difference. In terms of development, you probably all are familiar with the very long Vineland adaptive behavior scales. And what they do here, this is a fake example that I'm showing just to get familiar with the graph. On the horizontal axis is the age of the child or participant. In the vertical axis is what level of functioning they have. So a person with normal development would always intersect at the same point. So age 1, they would have a level of development equivalent to age 1. At age 18, real age, they should have a development of an 18-year-old. So we will see in yellow what happens with the participants with white Sutton. Each little blue dot is a specific participant. They are marked on the horizontal axis as their real age and on the vertical axis as how they're actually functioning. So what we see here is that language is compromised. So for example, we see all these dots, but we can also see that there's some people like up here. This is a person who's 17-year-old and is performing like a 12-ish-year-old. So the gap is not huge. However, not everybody behaves the same. There are some other people that reach adulthood and they function more or less at the level of a 5-year-old, and rarely there are people that remain nonverbal or very delayed. So again, it's a large spectrum. However, at age 4, 93% of all individuals are using language. In terms of personal care, 87% of individuals are likely to be feeding themselves at age 4, and some of them get sufficiently independent to also dress themselves and do more complex personal care skills. And socially, there are 67% of individuals that are making eye contact at age 4, and a good number of them also start making social friends, so in a more complex level of development. So in summary, the most common issues are intellectual disability and developmental delay, complications with language, some neurological issues. However, seizures are relatively rare, vision issues, and some gastrointestinal concerns. Less frequent are these, and in many cases, they're happier, more social than many other conditions with single gene alterations. So finally, if you are part of Simon Searchlight, I would very much encourage you to keep completing those forms every year. It's the only way we will know what happens long term. And looking at our survey completion rate, we are about, not specifically your community, but the more than 5,000 people in Searchlight, we're at about 70% completion rate of the surveys, and it's pretty steady over time. So the first thing we need to do is get people to start and then encourage them to stay engaged. It's the only way to know what will happen. If you by any chance know of anyone with some condition, they don't know what it is, they have not had a genetic diagnosis, let them know about us. We have a parallel study, Spark, that also offers whole genome sequencing for free, and return of results and genetic counseling, also funded by Simon's Foundation. So it will take everyone to get that knowledge. And the second thing is, last year, many people donated blood samples. Our plan until yesterday, I think, was that we would have a full team. It was 3 of us coming from Searchlight. We're bringing in phlebotomists and so on. But given airport problems, FAA, I'm the one who's in Oklahoma, so I was able to come, but everybody else could not make it. But in exchange, if you want to donate either a first sample or donate a new one, since we're running out of materials, please just sign up with Mandy and with Josh, and our team will reach out to you. We're going to offer you in-home phlebotomy, so we will schedule for a phlebotomy team to go to your home, do all the process, ship the samples to us anytime you want, because we're really, really sorry that we could not live up to our original commitment. So thank you very much.
So I have a question as far as the variant of insignificance, because both me and my daughter have the POGZ and the variant of insignificance. So I'm just trying to figure out, you said you could do more testing to figure that
The
part?
first thing we do is reanalyze your report. So see what the report says is the mutation, and run it again through all the new algorithms that are evaluating or defining what is a pathogenic mutation. So in many cases, it's not necessary to retest. We just need to look at that report again, and that's where we start. Are you already part of Searchlight?
I am part of both me and my daughter. Well, my daughter is, but
But you're the account holder.
I haven't seen a geneticist yet, but
We can work on that.
Just through my daughter's geneticist, that's how I found out.
Okay. Yes, we can chat about it, and then I can take down your information, talk to our genetic counselor so we can do a reassessment, and they'll reach out to you. And if it might be that it's not yet known, but we keep doing it every year. Once you are in the process of unknown significant, we will do it again next year if it's not this one. Okay.
Thank you for all your help and research that you're doing on this. And it seems like you have a very small pool to pull from. Is there other ways that we can help you get other people involved? You mentioned any rare disease and different things. What can we do to help, and the message that we can convey on our social media and different things we can do?
Well, one is through social media or the circles you move in, because I am aware of the fact that many of the patient advocacy groups, they go to meetings and they know other people in patient advocacy groups. So right now we have 185 genes on the list, but as you may all be aware, there are more than 1,000 genes already identified with autism and or neurodevelopmental disorders. And we're not close to adding more genes. If there are people that know their condition and they're not in the list for Searchlight, they can apply to have their gene included, and then we offer all the same set of things. So that's one way. The other one is if you have members abroad or you're aware of people abroad, make them aware of the fact that it is an international study, and that if they're uneasy completing surveys in English, we'll have 7 languages by the end of the year. So, because we have, I would say, close to 700 people in Searchlight that do not complete surveys because they did not mark English as their first language. And that is a big loss. So if you can promote that knowledge, that would be really helpful to us.
Thank you. Also, one other question as far as the future, you mentioned, coming up with some different pools put together and so forth. What do you see as a realistic timeline that, I hate to, you can't see the future, I get it, but just something to give some hope to some people in the room?
Future in terms of treatment, in terms of understanding of the condition. What is the question? To see if I can try to.
Most of us are dealing with the POGZ gene deficiency, and to do something to get, you see a lot of medical, breakthroughs. Do you see anything coming through along these lines?
I can't speak to POGZ directly, but I'll tell you where the field is moving. I think the main ideas I can share with you are that, one, the field is trying to listen to families, in that some families say, yes, of course we want a cure, but we are happy to get something that helps until we get to that place of the cure. And I think that is a new concept for many scientists. They are, jumping head first, and we want a cure. Well, yes, that sounds great. We all do. But is there something that we can do today, tomorrow, in 6 months that will not cure, but will alleviate some of the problems while we work on the cure? So that's a change in mentality. And there's a concept that is called drug repurposing. Drug repurposing is thinking of all the drugs that are already approved for anything. Think diabetes, think a headache, think an antibiotic. And that we know work for one thing, but that doesn't mean they have been tested for other things. What if there's something already out there that could be useful in a different condition? So there's a big push now to develop what are called high throughput platforms where you can test thousands of drugs simultaneously. And that's what those iPSC cell lines I mentioned are so important for, because you test those drugs on cells in culture that carry the exact variant of a person to see if it works. It's been very interesting. I'll give you a quick example. Metformin. Some of you may know it as a very inexpensive old drug for diabetes. Safe, inexpensive, well, no. Turns out metformin is being really helpful in autoimmune diseases such as lupus, which is a severe condition in many cases, rheumatoid arthritis, ulcerative colitis, and who would have thought it has nothing to do with diabetes? But it turns out that repurposing the drug is making a difference. So that's one path that I think is important to consider. If you can find a scientist that is willing to run your samples through that platform just to see, is there something that we could do in the meantime? That's one. Second one is the promise of gene therapy. That is a big jump. It is very expensive. There have been many hiccups along the road in getting to a therapy where you replace the gene or fix the gene efficiently, but it's coming along. There are some conditions that are starting to have some success. There are other ones that are probably not the best for your specific type of mutations that are called ASOs, which is a molecule that doesn't fix the gene itself, but that blocks negative effects of the abnormal protein. So there are different strategies, and many of them are actually happening in clinical trials in humans for these ultra-rare conditions. So we're so much closer than we were just 5-10 years ago. But at the same time, I would encourage you to explore all options, because you will try 10 and 9 will fail. So if you go one by one, it will take a very long time. So in this case, it's better to be broad than go deep. Once you find something that is promising, then dive in. But right now, don't say no to options. Just at least ask, try. Mm-hmm.
So I have a question. We went through the whole process. Someone reached out to us and said they grew some of those cell lines using my daughter's sample we provided at last year's conference. Is there a way, like somewhere in Switzerland, is there a way to find out what they're doing with them? Or
let me find out. We usually do not provide results on a one-to-one level, so to say, because doing that is unmasking the identity of the person. But we can tell you what the samples, in plural, are being used for. That we can do. Her name is Misha Kawanda, and she is one of the genetic counselors, the person that lives in Switzerland. Yes. So we can chat, you and I, a little bit later, so I can put you in contact with her and we can share with you what they're being used for in general.
I was just going to put a plug in for people that haven't completed the whole process. It really is easy. I mean, we donated at the last conference. They reached out about the cells in May, and it was just kind of an email exchange giving approval for them to release the cells to the researcher. And I just filled out, I think, the most recent set of questionnaires. So it really is super simple. So if you're intimidated by the fact that it might be a huge time commitment, it hasn't been for us. So.
And we're also trying to modify a little bit how we do it so that you can see how long time each survey takes to complete, and it won't be in a specific order. You will just see the whole list and you can say, okay, today I have 10 minutes. This is the one that's 10 minutes. I'll do it quickly. You get gift cards for completing surveys. Good timing before the holidays to get some nice gift cards. But yes, really the only way of getting more information is by strength in numbers.
Thank you so much for your time. I'm going to ask our community, please, if you donated last year, please sign up to have them come to your house. What a nice benefit that is, that they get to come to your house and you can give samples. If you didn't and you want to, please do that. If you're listening online, please reach out to us and we are happy to set that up. We would love for us to get more samples and more research going. Thank you so much for your time and for the knowledge and information and the time you spent to come here to do this. Thank you.